we'll learn about digestion and absorption of carbohydrates, proteins and lipids. So here the learning objective is to learn about the definition of digestion, to learn about the human digestive system, to learn about the digestion of carbohydrates, proteins and lipids, and also the students will learn about the absorption of carbohydrates, proteins and lipids. So in the previous session 8, I have described about the definition of digestion, types of digestion and the human digestive system. And digestion is the breakdown of larger insoluble food molecules into smaller water soluble food molecules so that they can be absorbed into the watery blood plasma. So the certain organisms, these smaller substances are absorbed through the small intestine into the bloodstream. So digestion is a form of catabolism that is often divided into two processes based upon how the food is broken down. It is differentiated or of two types. One is mechanical digestion, another one is chemical digestion. Mechanical digestion refers to the physical breakdown of larger pieces of food into smaller pieces, which is subsequently be assessed by the digestive enzymes. Whereas in chemical digestion, the enzymes break down the food into smaller molecules so that the body can use. This is the digestive system, which starts from the mouth, salivary gland, pharynx, esophagus, stomach, small intestine, large intestine, rectum and the anus. So the component of the digestive system consisting of alimentary canal and digestive glands. The alimentary canal includes the mouth, the tongue, pharynx, esophagus, stomach, small intestine, large intestine, rectum and the anus, whereas the digestive glands are the salivary gland, the liver, the pancreas, the gastric glands and the intestinal glands. If you just look on to the major digestive enzymes which is important for the human being to digest the compound molecules to simple molecules. As I told you that the polysaccharides, that is the carbohydrates, should be broken down to the simple sugar, that is monosaccharides and disaccharides and monosaccharides, that is the glucose, and the protein should be broken down into amino acids, and the lipid should be broken down to fatty acids and glycerol. So for this, the major important thing is the enzymes. So these enzymes, they act on starch or they act on the proteins or they act upon the lipids for the conversion into the simpler molecules. So the major enzymes are salivary amylase enzyme, the pepsinogen, the pancreatic alpha amylase enzyme, which converts the starch into disaccharide, the trypsinogen, which acts on protein to convert into peptides, chymotrypsin, elastase, carboxypeptidases, which converts large peptides into smaller peptides, and aminopeptidases, which is found in the small intestine, that is in the duodenum, which converts the large peptides into oligopeptides, and lipase, which converts triglycerides, fatty acid, glycerols into monoglycerides. And also, what are the enzymes that is secreted in the small intestine is enteropeptidases or enterokinase, which converts trypsinogen into trypsin, peptidases, which is secreted in the small intestine, that is in the region of jejunum, which converts the oligopeptides into amino acids, and disaccharides, which converts disaccharidases, which converts disaccharides into monosaccharides, and nucleosidase, which converts nucleotides to nucleosides and phosphoric acid, and nucleosidases, which converts nucleosides into sugar, purines and pyrimidines. So these are the major digestive enzymes which are secreted in the mouth and esophagus, pancreas, then the small intestine and large intestine, which are important for the conversion of the carbohydrates, proteins, lipids, and then lipids and nucleotides into smaller peptides. So these are the things which is important for the digestion of the food, the human beings. So let us come to the digestion of the carbohydrates. So this is the chart showing the digestion of the carbohydrate, how this carbohydrates, that is starch, which is a polysaccharide, is converted into simple sugar like fructose, glucose and galactose. So the digestion of the carbohydrates, when you come to the digestion of the carbohydrate, digestion of starch and glycogen begins in the oral cavity and it is completed in the small intestine. In the oral cavity, the food is mixed with the saliva and this saliva contains salivary amylase enzyme or ptyalin, which is also called as ptyalin, and this ptyalin is functionally at the neutral pH and it causes the hydrolysis of starch into three types of substances like small or limit dextrins, small or limit dextrins, maltose and isomaltose. So the sweet taste of some of the food after chewing is due to the maltose. About 30% of the food starch is hydrolyzed in the oral cavity and the process continues even in the stomach till the gastric juice mixes with the food. The gastric juice does not contain the carbohydrate digesting enzyme. So in the duodenum, the food mixes with the pancreatic juice and this juice contains an amylase known as pancreatic amylase. So this, like a salivary amylase, it acts on the starch and dextrin to produce limit dextrin, maltose and isomaltose. So the intestinal juice contains isomaltose and maltase enzyme for hydrolyzing the limit dextrins, isomaltose and maltose into glucose, and glucose is absorbed in the small intestine, and there are two more carbohydrate digesting enzymes in the intestinal juice and they are called as sucrase and lactase, and this sucrase hydrolyzes sucrose or common sugar into glucose and fructose. So you can see here the sucrase which converts the sucrose into glucose and fructose, and lactase which converts lactose into glucose and galactose. This is how the digestion of the carbohydrate occurs in the human being. Did you understand? Next is the digestion of the proteins. Before coming to the digestion of the proteins, we have to tell about the digestion of the fat. So that is the fat digesting enzymes are also known as lipases and they are present in gastric juice, pancreatic juice and intestinal juice. So these lipases are water soluble enzymes while fat and oils are insoluble in water. You know that fat and oils are insoluble in water. Therefore, these lipases, they act only in the region of water-fat interface. So in order to increase the interface, fat have to be broken down into fine droplets through the process what is called as emulsification of fat. So this is done by bile salts like sodium trochrate and sodium glycolate, and in the stomach there is no fat emulsifying agents. Therefore the gastric lipases is not much functional in the small intestine. So both pancreatic lipases and intestinal lipases are active after the bile salts produces the stable fine emulsification of fat. Emulsification of fats. So the pancreatic lipases hydrolyzes the fats in the fats which changes from triglycerides to biglycerides and biglycerides to monoglycerides, a simple form. So fatty acids are released in both the steps and the intestinal lipases causes further hydrolysis. As a result, fatty acids and glycerols are produced. The simple substances where the lipids is converted into fatty acids and the fatty acids and the glycerols. So this is the step what happens in the digestion of the fats. So you have to remember how the digestion of fat occurs, how it split into the lipids into fatty acids and the glycerol, and what is the step that is called as, what is the terminology that is called as emulsification of the fat. So you have to understand about the digestion of the fat. Coming to the digestion of the protein. So as I told you in the digestion of the proteins, the protein has to be converted into simple amino acids. So the digestion of the protein, here the saliva does not contain any enzymes for the digestion of the protein. So the gastric juice contains hydrochloric acid and the pepsinogen and prorinin in case of calves. This hydrochloric acid makes the food acidic and it also changes the proenzyme, that is pepsinogen, to pepsin. So this you can see the pepsinogen is converted into pepsin with the presence of an hydrochloric acid that is secreted in the stomach, and then pepsin at the pH of 1.2 to 1.8. This pepsinogen is converted into pepsin. Okay. Then the proteins in the presence of pepsin, it is converted into peptones. Okay, and it also changes the proenzyme pepsinogen and prorenin into active enzymes pepsin and renin. So this is also important step that acts in the digestion of the protein. And also the renin, which take part in curdling of the milk by converting the casein into paracasin, and that combines with the calcium. The casein, that is also called as a milk protein, in the presence of HCl and pepsin and renin enzymes, which converted into paracasin, and this paracasin which combines with the calcium to form calcium paracasinate, and calcium paracasinate is also converted into simple molecules that is called as peptones by the presence of an enzyme what is called as pepsin. Okay, this is the important steps that you have to remember for the digestion of protein. Next is the bile that does not contain any action of proteinaceous food except that helps in making the food alkaline. Here, the pancreatic juice with the pH of 8.8 contains the proenzyme proteinaceous substances, that is trypsinogen, chymotrypsinogen, pro-carboxypeptidase, and pro-elastase. So this trypsinogen is changed into active enzymes trypsin by ingredients of intestinal juice, enterokinase or enteropeptidases. Here you can see the enterokinase or enteropeptidases. This trypsinogen is converted into trypsin. Okay, then the trypsin coagulates blood by changing the fibrinogen into fibrin. So here you can see by the help of an enzyme trypsin, the fibrinogen is converted into fibrin. Then the chymotrypsinogen is converted into chymotrypsin by the help of an enzyme what is called as trypsin. Okay, so that pro-elastase to elastase and pro-carboxypeptidase to carboxypeptidase, and it hydrolyzes the proteins into larger peptides. This is the thing which happens in the digestion of the protein. And then you can also see there is casein with the help of an enzyme chymotrypsin is converted to paracasin. Then paracasin with the help of when it combines with the calcium to form calcium paracasinate, and paracasinate is converted into peptones as I told in the previous slides, and proteins and peptones with the presence of an enzyme trypsin and chymotrypsin is converted into peptides, and peptones in the presence of carboxypeptidases is converted to peptides and amino acids, and the elastin in the presence of an enzyme elastase is converted into peptidase. So this is because of intestinal juice which is alkaline and 8.3 pH, and this has two proteolytic enzymes, namely aminopeptidases and dipeptidases. They act on peptides, and these peptides in the presence of an aminopeptidases is converted into amino acids plus smaller peptides, and smaller peptides, that is dipeptides, is converted into amino acids by dipeptidases enzymes. So when you write in the examination about the digestion of the protein, you have to go through this major important step that acts in the conversion into smaller molecules, and then you have to add this chart also in order to write the explanation on the digestion of the proteins. When you write this write this symbol along with the presence of an enzyme, how it is converted into simple molecules, so that you can write by yourself the note for the digestion. And now you have studied about the digestion of the carbohydrate, which are the enzymes that are involved in the conversion of the polysaccharides into monosaccharides, and then you have also studied how the digestion of the lipids occur and what is emulsification means, and the lipids, how it is converted into fatty acids and glycerol, and also you have also studied about the digestion of the proteins, the enzymes that are involved in the digestion of the proteins, how it converts into simpler molecules. And after studying about the digestion of the proteins, digestion of the carbohydrates and digestion of the lipids, let us come to the absorption. So what is meant by absorption? So it is nothing but the passage of digested material, nutrient and inorganic substances from the lumen of the alimentary canal into the blood vascular system for the distribution to different parts of the body. So after the digestion, the next process is absorption, so that is absorbed through the blood and it is circulated or distributed to the different parts of the body, so that we will get the energy to do the work. So major of the absorption occurs in the small intestine and it is followed by the large intestine. Okay, then when you look on to the, as I told you that major of the absorption occurs in the small intestine and it is followed by the large intestine. So when you come to the first region, that is in the buccal cavity, there is as so much nutrients is absorbed in the buccal cavity because absorbable substances are not produced in it. So however, some drugs and water are known to be picked by the mucus from the sublingual area. So absorption in the stomach, in stomach the digestion is incomplete. So absorbable products are formed on the wall of the stomach. So they are known to absorb water, sugar, alcohol and some drugs in the region, that is in the stomach. When you come to the small intestine, the digestion is completed in the small intestine, and the small intestine is also a major area of absorption and it receives about 8 to 9 liters of liquid food. And here 80% of the absorbed in small intestine and remaining about 1.5 liters of the food passes into the large intestine for the further absorption and conversion into fecus. So here the small intestine has an internal surface due to the presence of three types of structures, that is called placa circularis villi, microvilli, and the absorptive area is increased by 1000 folds. The absorptive capacity is more than 10 times as actual absorption. Okay, carried out by the small intestine. Absorption occurs by the following method, that is diffusion, that is osmosis, facilitated diffusion, active transport and co-transports. And the absorbed materials are passed from the enterocytes to paracellular spaces, from where they are picked by the blood capillaries and lymph vessels. This is a section of the intestinal villi. So the villi helps in the absorption of the digested food. As I told you, the absorption of sodium, the absorption of the glucose, absorption of the calcium and water by the intestinal epithelium, and as I have told, by the different methods the absorption occurs. So the water absorbed through the process what is called as an osmosis, and sodium by active transport, glucose is absorbed into the intestinal epithelium by what is called as a co-transport, and calcium is absorbed into the intestinal epithelium by the diffusion, and the absorption surface is increased by the presence of one microvilli. So the villi helps in the absorption of the food materials. So this thing you have to understand when you come to the absorption of the food. Okay, as I told you that the absorption, how the absorption of water occurs through osmosis, and absorption of ions by the active process, and then not only the sodium is absorbed, the other ions are also absorbed, that is calcium ions, potassium, magnesium, phosphate, and other ions are also absorbed through the active process, and absorption of calcium is controlled by vitamin D and parathormone. Next, absorption of monosaccharides, the glucose enters into the intestinal epithelium along with the sodium. So the mechanism is what is called as co-transport. So through co-transport the glucose is absorbed, and then as a part of glucose also enters into the paracellular spaces directly through the cell junctions, and it seems to be required for developing high osmotic potential and this osmotic entry of water, and the galactose is also absorbed by the intestinal epithelium by the method similar to that of the glucose, that is post transport of sodium, and fructose is also absorbed through the diffusion, however, inside the enterocytes, and it is phosphorylated and changed to glucose, in which form it is passed on to the paracellular spaces. And when you come to the absorption of the proteins, the amino acids, most of the amino acids are absorbed through sodium post transport mechanism and few picked up through facilitated diffusion. And also the absorption of glucose, sodium, calcium and water by intestinal epithelium. And when you come to the absorption of the fat, the bile acid micelles carry digestive product of fat over the surface of the microvilli of enterocytes, and the digestion product is monoglycerides, fatty acids, etc. passes through the membrane by the process what is called as diffusion. Okay, then when you come to the this structure, when you draw the structure or when you draw this picture, that is absorption of the digested food, that is amino acids and monosaccharides is absorbed by the process what is called as facilitated transport or co-transport. And as I told you, the fatty acids is absorbed by the simple process called as diffusion that takes place in the lumen of the small intestine, then into the epithelial cells of the intestine, then carried over to the blood capillary. So this is the sequential process how occur from the lumen of the small intestine, how protein is broken down into amino acids and carbohydrate is broken down into monosaccharides and bile salts, that is fat globules, that is triglycerides is converted into simple fatty acids, that is monoglycerides by the process what is called as emulsification. This smaller piece what is called as emulsified droplets with an enzymes what is called as a lipases, which is converted into smaller fatty acids and glycerols, and how they are absorbed by the epithelial cells of the intestine, and then how it is transported into the blood capillaries so that absorption of the digested food occur. And formation in the large intestine. So absorption, that is absorption in the large intestine. Formation in the fecus, about 1.5 liter of chyme or slurry containing the indigestible material passes through into the large intestine, and first half of the colon contains the number of bacteria, example E. coli, Streptococcus faecalis. Some of them are symbiotic and produces vitamin K, thiamine and riboflavin and vitamin B12, and this bacteria feeds over the entire indigestible matter so that it gives out gases like carbon dioxide, methane and hydrogen sulfide. So the small amount of cellulose may be digested, and in the absorption of the colon, the vitamin released by bacterial flora, sodium and chloride ions are passed into the epithelial cells. Absorption of sodium is active process, and the other absorbed through diffusion. Sodium and chloride ions create osmotic potential across the colon epithelium, which facilitates the absorption of water. As a result, emulsion of indigested matter present in the colon is changed into semi-solid residue called fecus will become toxin when it is present for a longer time. So this has to be eliminated. That is eliminated. And this fecus represents a solid waste matter. It is also called as a stool, which is formed by indigestible matter of food. After the latter, it has been acted upon by digestive enzymes. The adult forms about 150 grams of fecal matter is thrown out, and it contains about 65 to 75% of the water, while the remaining 25 to 35% solid matter. And fecus is brown color, and the color is due to the strecobolin and urobilin, which is formed by the bilirubin by the bacterial action. So this color is brownish. And the bacterial also provides the characteristic odor to the fecus, and the various odoriferous ingredients are hydrogen sulfide, methane, indole and mercaptan. So the solid matter of the fecus is composed of 30% of indigested roughages and unused digestive juices, and 30% of dead bacteria, and 10 to 20% of the fat, to 3% protein, and 10 to 20% inorganic matter. So egestion is the last process. So it is the elimination of fecal matter from the body, and it is also called as defecation. So this is what you have to write, study in the session, that is digestion and absorption of carbohydrates, proteins and lipids. Let us go to the multiple choice question. Saliva contains an enzyme called ptyalin, proteases, sucrases, lipases. The answer is ptyalin or salivary amylase enzyme. The next question is, absorption of glycerol fatty acids and monoglycerides take place in lymph vessels within the villi, wall of stomach, colon, sodium glycolate and sodium trocolate. The answer is lymph vessels within the villi. So villi is important for the absorption of monosaccharides, glycerol and fatty acids, etc. Next, what is the function of the trypsin? So the function of the trypsin is breakdown of carbohydrates, breakdown of fats, breakdown of proteins, manufacture of the proteins. As I told you in the previous slides, the main function of the trypsin is the breakdown of proteins. Next, coming to hydrochloric acid present in gastric juice converts pepsinogen into pepsin, polypeptides to peptones, disaccharides to monosaccharides, proline into renin. So the answer is pepsinogen to pepsin. If you want the further information, you can refer these books. So what it is given in the references. Thank you.